Hello everyone and welcome to our next lecture section. Today we're going to be discussing chapter 6, which reviews microbial nutrition and growth. So all organisms have to have some kind of basic nutritional needs, just like even you and I do. We have to consume food to meet our nutritional requirements. Well, microbes are the same. They also have to have some kind of essential nutrients that they have to consume. And as a matter of fact, that is the definition of our essential nutrients. They're things that must be consumed because we cannot make them. In general, there are several different atoms that are associated with the chemical makeup of these essential nutrients that are listed here. But how they get these elements in the form of nutrients varies from organism to organism. And these various nutrients can be classified in two ways: how much we need and how much carbon content they have. So let's look at categorizing nutrients first according to the amount needed. So there's two basic classifications when we are talking about nutrients in terms of how much we need, and they are as follows: macronutrients and micronutrients. So when we say macro, that means large. So we need lots of these macronutrients, whereas micronutrients or trace elements are needed in small quantities, hence the term micronutrients. So our macronutrients are going to contain lots of carbon, hydrogen, nitrogen, and oxygen. And if you took biology with me, you might recognize that as the mnemonic device I give y'all in Bio 1 as CHON. All right, CHON, our macronutrients, the things that we have a high need for, are going to be composed of the elements carbon, hydrogen, oxygen, and nitrogen. This table here from your book illustrates that. So if we look at the title, this is the chemical composition of an E. coli cell, which is a bacterium. So if we look at the four most prevalent elements, we have carbon, hydrogen, oxygen, and nitrogen, CHON. The trace elements are found in much smaller quantities, as you can see here. You do not need to memorize all of these, but I would recommend knowing CHON. So that's categorizing nutrients based on the amount we need. Now we're going to move to categorizing them based on their carbon content. So we can classify nutrients as organic or inorganic nutrients. Organic nutrients will always contain both carbon and hydrogen atoms. And these are typically products of living things. Some examples are listed below. Methane, which is a gas, CH4, it has both carbon and hydrogens. If we were to analyze the structure of a carbohydrate or a lipid or a protein or a nucleic acid, all of those basic compounds that make up living things are going to have carbons and hydrogens. Inorganic nutrients are a combination of elements that contain something other than carbon and hydrogen. So for example, water, H2O, it contains hydrogen, but there's no carbon, so it is considered inorganic. Likewise, carbon dioxide, listed here, contains carbon, but no hydrogen, so therefore it is classified as an inorganic molecule as well. And these inorganic molecules of various types are found throughout nature. They're found in the Earth's crust, bodies of water, in the atmosphere, gases such as the carbon dioxide we listed. Oxygen would be inorganic because its chemical formula is O2, no carbon. And metals and their salts would also be considered inorganic nutrients because they do not contain carbon and nitrogen, or I'm sorry, hydrogen, carbon and hydrogen. So now let's look at the chemical analysis of the microbial cytoplasm. So as we should be familiar, if we have this is my bacterial cell, inside of the cell there is cytoplasm. That's the fluid within the cell that the various organelles are suspended in. If we were to analyze that cytoplasm, 70% of it is water. Right? You need to know that, big deal here. 70% of the cell is water. Now, if we were to take water out of the picture here, pretend we sucked out all the water and so all we have left is like the dry components, then 97% of what's left over besides water are organic compounds, like those proteins, nucleic acids, carbohydrates, and lipids. And again, that means that all of them have C and H, among other things. So if we look at this chart here from your book, you do not need to memorize the charts, but you should know water is the primary component. 97% of the dry weight is organic compounds, such as these that are listed. You don't need to know the specific numbers, like you don't need to know 50% of the dry weight is protein, 20% is nucleic acids in the form of RNA, and so on. You just need to know this list here. But I'm just showing you where it comes from your text. And these organic compounds are made up of carbon, hydrogens, and amongst other things, most of which is CHON, as well as P and S, which stands for phosphorus and sulfur. All right, so most chemical elements are available to the cell as compounds, not as pure elements. So I just wanted to point that out. It's not like there's individual carbon atoms that are floating around in the cell. It's just one of the elements that are making up a organic compound, like a protein or a lipid or a carbohydrate or a nucleic acid. All right, I know this table looks a little intimidating at first, but we're going to work through it. Our next topic of concern is to describe nutritional type. So how we get these various elements or organic compounds like the proteins, nucleic acids, etc., is based on an organism's nutritional type. And so this is a listing here by category of all the various nutritional types concerning microbes. So you'll notice there is autotrophs and heterotrophs, and then there's subcategories of those as well. But I want to kind of point out a couple of things that are consistent in the chart that might help you to be able to master it better. So if we'll notice, all autotrophs have a carbon source of carbon dioxide. All heterotrophs have an organic carbon source. All there's a photo in the beginning, then it means the energy source is sunlight. So those are just some consistent patterns in the chart that I think are of relevance. Also, anything that's a chemo is pertaining to chemicals, and we'll get to the specifics as we go one by one. You do not need to know specific examples, but you should know this here. If I were to ask which of the following would describe the nutritional type of a microbe that uses carbon dioxide as the carbon source and sunlight as the energy source, then you should be able to tell me a photoautotroph. So some rules of thumb: the microbe's carbon source and energy source determines its nutritional type. Hence, that's why they're labeled here at the top as categories in this table. Our carbon sources can be heterotrophic or autotrophic. Our heterotrophs must obtain its carbon from an organic form. Whereas autotrophs, they convert inorganic carbon dioxide, a gas, into organic compounds. We call these self-feeders, hence the name autotroph. So these are not nutritionally dependent on other living things because they can make their own food. Like, for example, this isn't a microbe, but plants. We're very well aware that plants make their own food. So they are autotrophs, and more specifically, photoautotrophs, because they get their energy from the sun. All right, so their energy source is another category here that determines nutritional types. Phototrophs are organisms that can photosynthesize. So they get their energy from the sun. I'm terrible at drawing with this thing, clearly. Chemoautotrophs get their energy from chemical compounds. So again, in summary then, our photoautotroph will gather energy from the sun, and they will harvest carbon dioxide as a carbon source to produce the food that it needs. A chemoautotroph will utilize chemicals that will allow it to self-produce food. So for example here, chemo-organic autotrophs, they use chemicals, that is their energy source. The energy source specifically is organic chemicals, hence the name chemo-organic. Auto means that we have our carbon dioxide as a carbon source. Chemo-lithoautotrophs, so the difference here being litho instead of organic. So they're still going to use chemicals as an energy source. Specifically, it's going to be inorganic chemicals. That's where the litho comes from. Auto, again, meaning that it's going to have CO2 as its carbon source. Moving down to our heterotrophs, all of these have a carbon source that is organic. Photoheterotrophs will have an energy source of sunlight. Chemoheterotrophs will have an energy source of chemicals, and we have two subtypes, our saprobes and our parasites. The difference being our saprobes are going to be metabolizing organic matter of dead organisms, but parasites utilize a live host to gather food. All right, so again, we'll go through this one more time, and I have this part color-coded, so if you prefer a color-coded explanation like this, then that might be the best way for you to study this, but if you like tables, I'm a table person, so I included that table in the PowerPoint as well. So again, we have our autotrophs, and they are going to be classified as either photoautotrophs, and we have chemoautotrophs. The photo here means sunlight. It uses the sun for energy. Auto meaning CO2 is the carbon source. So if we look at both of these down here that are chemoautotrophs, you'll notice that they are autotrophs and utilize CO2 as a carbon source. Auto, CO2 is carbon source. However, chemoorganic autotrophs will utilize organic compounds for energy. Chemolithoautotrophs use inorganic compounds for energy. All right, so I accidentally included the picture again, so I'm sorry about that. We're not going to go through it another time, but it's the same exact picture as the one I showed previously. All right, so now looking at heterotrophs and their energy sources, the photoheterotrophs, photo meaning again it uses sunlight for energy, hetero meaning it has to be able to use some kind of organic materials as its carbon source. Chemoheterotrophs can be further classified, just like we were talking about earlier, into saprobes and parasites, and it's going to be dependent on whether they utilize either dead or alive, dead organic matter or a live host as a food source. And both of them, since they have the term hetero in there, will utilize organic materials as their carbon source. Okay, so now moving to 6.3, this section discusses a process called binary fission, and I don't want to spend too much time worrying about the details of binary fission, but you should know that it is how bacteria divide. It's how they multiply. So if you took either biology or A&P, which you all have taken one or the other, y'all should have talked about a process called mitosis that we see in eukaryotic cells. In prokaryotic cells, this process is called binary fission, and there's a lot of similarities in regards to binary fission versus mitosis. There's a lot more complexity in mitosis, But essentially, to keep things very simple, we have a cell here. This is a bacterial cell. You can see the pink is representing its DNA, and all bacterium have a single circular loop of DNA as shown here. And it's really long. You know, think about this like a really, really big rubber band. It's a single closed loop, but you can kind of squish it all and pile it into an area like this. So that pink is representing the DNA of this bacteria. So in order for this process of binary fission to happen, it has to replicate. All right, so basically you can think of this like a copy and paste function. So if we take this chromosome and duplicate it, we now have 2. So here you see there is 2 chromosomes now, and they are genetically identical because it is a copy and paste type of action that causes that replication event. So you now see that the cell starts to elongate. Eventually there's a set of proteins that will form this area in between called the septum of the cells that are dividing, and that's going to serve as the plane of division. So you can see here it's getting smaller, and it will essentially pinch off sort of to eventually separate 2 new cells. So when that septum is complete, the cells are now divided, and we now have 2 cells that are genetically identical. So in review, we start off with 1 cell, DNA is replicated, the cell divides, and now we have 2 cells, and these 2 cells are genetically identical to the original one, and of course they're also then directly genetically identical to one another. So now if a second round of division were to take place, then we would have 2 cells that stemmed from this one and 2 cells that stemmed from that one, and we would have a total of 4 genetically identical cells, and the process can continue. So the reason why we needed to briefly go over that is because our next topic is about population growth in bacteria. So the process of creating new bacteria cells, that division, that binary fission process, is going to result in double the population each round of division that occurs. So we call this the generation time or the doubling time. Know these terms. And that is the time required for a complete binary fission cycle. So each generation increases the population by a factor of 2. And again, that's because if we start off with 1 cell, it divides, we have 2 cells. If these 2 cells divide, we now have 4 cells. If all of these cells then divide again, we now have 1, 2, 3, 4, 5, 6, 7, 8 cells. So every time we have a round of replication, we are doubling the population from the previous generation. So this would be Gen 1, Generation 2, 3, 4, and each time it doubles. And as long as the environment remains favorable, that doubling effect will continue at a constant rate. And for that reason, we can use math, yay, to predict population sizes. And there's an equation that goes with it called the theoretical, and that's in quotations for a reason. I'll explain why in just a second. But it is the theoretical mathematics of population growth. And the formula for that is n sub t equals n to the second power of n to the, or a second power, I'm sorry, I misspoke. N sub t is equal to n times 2 to the nth power. There we go. It was a little tongue twister. Sorry about that. But what do these numbers then stand for? N sub t is the total number of cells in a population. So T represents this population number at any point in time. The N represents the starting number of cells, and the exponent little n denotes the generation number, And collectively, 2n represents the number of cells in that generation. So I have some good news for you. I'm going to show you an example of how you could apply this calculation, but I'm not going to ask you to do it on the exam, and I'll explain why momentarily. But I do think it's of interest, so we'll spend just a second reviewing how this would work. So let's say that an egg salad sandwich was left in a warm car for 4 hours, and it had 10 Staphylococcus aureus bacterium on it to begin with. And we want to know how many of these bacteria are going to be on the sandwich after these 4 hours of it sitting in the car. And it says the generation time for Staph aureus is 20 minutes. So here's our equation again. We have n sub t equal to n times 2 to the nth power. So first thing is we would want to incorporate the generation number because it was given, as well as n, which was also given. So what we're trying to find here is n sub t, and it is equal to n, which we know is 10 because we started with 10 of these Staph aureus bacteria, multiplied by 2 to the nth power. That n superscript represents the generation number. To calculate the generation number, we would take the time that it has been growing and divide it by the generation time, which was given. So it says that 4 hours is how long it's been growing. If you convert that to minutes, it's 240 minutes divided by the generation time, which is 20. So if we do 240 divided by 20, we get 12. So that's our little n. So now we have our final number, n sub t, which we're trying to find, set equal to 10 times 2 to the 12th power. And so if you do that calculation, you're going to get 40,960 bacteria after 4 hours when we only started with 10. All right, so again, you won't need to do those calculations on the exam, but I had a point in showing you that. It is to show you how quickly bacteria can reproduce, and they reproduce exponentially, meaning every generation it doubles. So this is similar to the picture that I drew for y'all on the other slide, but if we start with 1 cell, then we get 2 after the first round of division, and after a few more rounds of division. So this is 1 round, 2 rounds, 3 rounds, 4 rounds, 5 rounds. We have 32 bacteria here after just 5 rounds of division. So if we start off with a higher number of bacteria, then that number is going to drastically increase compared to this specific example here. Like say if you had 10 as your starting number, well then after 1 round of division you're now going to have 20, and so on. So very fast population growth in these bacteria theoretically. Now I keep saying theoretically. Why do I keep saying that? So while theoretically we can predict growth with this mathematical process, it's not really always the reality, because there's lots of various factors that can limit population growth. So instead, what we typically see is a predictable pattern of bacterial growth that we call a growth curve that looks like this. So what we're seeing in this chart here, or graph, excuse me, is after so many hours, the number of viable cells that are in this hypothetical test tube. And it's even color coded, which is pretty neat, showing you that each one of these test tubes, which has some bacterial culture in it, is going to have live cells, mini live cells, or they're going to have dead cells. So in the beginning, if I take a test tube and I have some kind of nutrient broth in it, and I culture some bacteria of whatever species, let's say it's E. coli, then in the very beginning, we're going to have a very few number of live cells. And this is called the lag phase. And then what we're going to see is over time, the first several hours here, we're going to see exponential growth to where we see many cells eventually that are living. We'll move on. After more time, there'll be a stationary phase where you'll see the bacterial number will kind of level off because there's some dead cells now, but there's also a lot of live cells. And then eventually, over more time, we'll enter into the death phase where we'll see even more dead cells relative to the number of living cells and a decline therefore in the cell population. So let's go over each one. The lag phase. This is a flat period of growth. That's the very beginning when we first cultured this hypothetical bacterium in this test tube. So these newly inoculated cells, they kind of require this like adjustment period before they really start to proliferate. So the cells are not yet multiplying at their maximal rate, so the population of living cells is pretty sparse. Very few living cells at that point. Moving to the second phase, we're going to see exponential growth next. So exponential growth is that doubling effect that we said was theoretical. So we do see that as part of the growth curve, but it's just not limitless like we described it in the mathematical sense. This exponential growth will occur as long as the cells have enough nutrients and they're happy and the environment's favorable and they have no pressure on them and thus no limited resources essentially. So no different than if you have a population of a bunch of let's say a bunch of lions. There's only a limited food source. So the population is going to reach its maximal capacity at some point and then some will die. So just like that, it applies to bacteria. We'll see exponential growth during the time periods where the environment is favorable and nutrients are plentiful. But as nutrients are used up, then we're going to see this leveling off. This the capacity, the maximal capacity for the cells are at this point. So we're seeing the stationary phase next. The stationary phase is where we're going to have cell birth rates and death rates being equal. So for every time a cell divides, there's going to be cells that die at equal rates. So this is caused again by an unfavorable environment. There's a depletion of nutrients, a depletion of oxygen, there's excretion of biochemical waste into the growth medium that is toxic for these bacterial cells. So we're going to see a leveling off of the number of cells that are alive in this portion. And then eventually we see the death phase. Now you see the population number is declining. They begin to die quicker and quicker because of the buildup of waste, and so we don't see a whole lot of new cell division. It's more death than it is binary fission. So we're going to see eventually these cells are going to be depleted in numbers. All right, so that is our typical thing that we would see in a cell or microbial population growth over time. Now, the last thing we're going to talk about is some population growth techniques, how we can observe it, because we already said that using that mathematical formula is not the ideal way to calculate this. And again, it's because it doesn't take into account all of the various pressures that microbes can have in this laboratory condition. So in general, the steps for doing this technique, which is called a variable plate count, it requires culturing. So to culture a bacterium, you're going to inoculate, in this case, a broth. So remember in chapter 2 in lab, y'all talked about the difference between broths, which is a liquid medium versus agar plates and so on. So we have a broth that we're going to inoculate. We're going to incubate it, and then we're going to sample the broth at regular intervals. And that sample is going to be used to create a spread plate, and then we're going to count the colonies that grow after that plating. So let's go through this picture from your text. So what it's showing you is that initially we have a broth that is inoculated. So this is an inoculated flask that has been inoculated with a bacterium. So we're going to take 0.1 at 60 minutes. 60 minutes. We're going to take 0.1 milliliters of this broth, and we're going to add it to a test tube. At 120 minutes, we're going to do the same thing. We're going to add 0.1 milliliters to test tube number 2. At 180 minutes, we're going to take 0.1 milliliters and add it to test tube number 3. And then in test tube 4, we're going to wait until it's 240 minutes since we inoculated it, and we're going to put 0.1 milliliters into that tube, and so on and so forth. So I hope that at this point you should expect, without even looking at the picture, that we will see more bacteria accumulate in this inoculated flask the longer it sits there, because initially we have the lag phase and then the exponential phase. Now eventually it will die off, like we saw previously in that growth curve. So, but this is only showing you up to 600 minutes. So for each one of these test tubes that we inoculated over time, we will then plate them onto agar plates. And then we can count the number of colonies. And each of these spots that you see on the plates are a colony. It's a CFU, a colony forming unit. So it's representing the CFU because each colony is basically a mound of bacteria that began as a single bacterium. So if I look at my first sample, I plate it onto this agar plate, well, I didn't see any bacteria because there's so few in this initial inoculated flask. We just happen to not have any at the 60-minute mark. But if I wait some additional time, another hour, and I took that sample and plated it, now we see 2 CFUs, 2 colony forming units. That means on this plate there was 2 bacteria, and after incubation we could see them because these colonies are basically mounds of bacteria that derive from the one bacteria as it was as we made the plate. So this was one bacteria that now has a colony of them, and this colony derived from a different single bacterium. So as we move forward, 180 minutes, now we're seeing 4 CFUs, and then 7, 13, 23, 45, 80, 135, and 230. So I did not count those, obviously. That's listed luckily for us in this nice picture. But so what we're seeing here is an increase in the number of our colony forming units. So thus we must have an increase in number of microbes over time. That's our exponential phase that we've been talking about in the past few slides. Now, if we were to continue doing this well past 60 minutes, let's say for a week, at the end of that week, we would see very few colonies remaining that would match this chart. In the very first part, we had very few colonies. Then we had exponential growth where we see many colonies, more and more and more of them. Then we would see the stationary phase after, let's say like 5 days. And then eventually after 7 to 9 days, we'll start to see less viable colonies. Okay, so there are other methods that we can analyze population size without culturing, right? So just to understand the difference here, we needed to culture bacteria and incubate them over time to do this process, and it was pretty lengthy. So we can utilize another method that's simpler that can analyze the population size, and it's the turbidity or turbidometry method. And so what it analyzes is how turbid a substance is. So a clear nutrient solution, like the one seen here on the left, will become turbid or cloudy, that's what turbid means, as microbes grow in it. So the greater the turbidity, the more cloudy it is, the larger the population size of bacteria. And these are measured using a fancy machine called a spectrophotometer. So what it does is it analyzes the amount of light that is allowed to pass through the tube. So if I have this clear, low turbidity in this broth sample here, and I shine a light nearby it, there's a little light in the machine, and what it does is it shines, and then there is a receiver that records how much of the light was transmitted through this broth. And so in this scenario, since there's low turbidity, it will have a high transmission rate. which will mean there is a low amount of bacteria. On the other hand, here we see a high turbidity sample. So if this is my sample and it's very turbid because it has all of these solid particles of these bacteria within it, then that light is not going to be allowed to fully transmit like it did in scenario 1. So it will measure less transmittance, which would equivalent to having a higher concentration of bacteria in the sample. All right, so there are a few other examples of how we can calculate population size. We can do it by counting, probably the most pain in the butt way to do it, to be honest, because you can directly measure them microscopically. There are cytometers, but this will do, it will have it's a slide that you put on a microscope and it has all these little lines on it. We're not going to go into a lot of detail, but basically they will come with instructions based on how it's made to give you calculations on how, if there is this many bacteria in this square, then how many are there per microliter of your sample. But we're not going to go to the details, but you'd have to individually count these bacteria on this microscope slide. There's a culture counter. These are pretty neat. They electronically scan a fluid as it passes through this little tiny pipette, so it can utilize electrical signals to count the cells. Flow cytometer is pretty popular. It works similarly, but it can measure cell size on top of counting them, so we can differentiate between live or dead cells or even cell types. Genetic probing is another example where we can count the cells. It uses PCR, and PCR stands for polymerase chain reaction, to quantify bacteria. So we won't go into any more details on PCR and real-time PCR. So we'll get to that a little bit later, possibly when we do our biotech stuff. All right, so that is chapter 6.